Hi, let us discuss the high-yield questions from general microbiology. Let us move on to the first question. Which of the following is not Coates' postulate? Your options are: a microorganism should be constantly associated with lesion of the disease. It should be possible to isolate the bacterium in pure culture from the lesions. Inoculation of pure culture into laboratory animals should reproduce the lesions. Administration of broad-spectrum antimicrobial agent eradicates the organisms and cures the diseases. So of all the four options, the answer over here for this question is your option D. So now how did we arrive at this? Let us have few discussion on Robert Koch. So Robert Koch has contributed a lot to the field of microbiology. So what are his contributions? So basically he is the one who used solid media for culture of bacteria, and he also introduced methods for isolation of bacteria in pure culture. And to describe the motility, he described hanging drop method for testing the motility of the bacteria, and he discovered bacteria like cholera, anthrax bacilli and tubercle bacilli. So you remember it with a mnemonic CAT. So cholera bacilli, anthrax bacilli and tubercle bacilli. And he gave Coates' postulates. So what are Coates' postulates? So first of all, it states that the microorganism should be constantly associated with the lesions of the disease. Second, it should be possible to isolate the organism in pure culture from the lesions of the disease. The third postulate is inoculation of such pure culture into laboratory animals should produce lesions. And finally, it should be possible to re-isolate the organism in pure cultures from the lesions produced in laboratory animals. So these are the 4 points he gave. So this is Coates' postulates. So he describes a microorganism can be accepted as the causative agent of an infectious disease only if following conditions are fulfilled, so which is called as Coates' postulates. Now, is there any exception to Coates' postulates? Yes. So the exceptions to Coates' postulates are, you remember it with the mnemonic my nest. So you remember it as my nest. So normally we write nest as NEST, but here you are going to add I. So my nest. So my stands for Mycobacterium leprae, and nest stands for Neisseria gonorrhoeae, and T stands for Treponema pallidum. So you have 3 organisms which are exceptions to Coates' postulates. Now moving on to what is Coates' phenomenon. So we have heard about Coates' postulate. So what is Coates' phenomenon? So now normally what is Coates' phenomenon is, now Robert Koch he observed that guinea pigs which is already infected with tubercle bacillus, they developed a hypersensitivity reaction when they were injected with tubercle bacilli or its protein again. So this reaction of hypersensitivity to the tubercle bacilli is called as the Coates' phenomenon. So Coates' phenomenon is nothing but where the guinea pigs already infected with tubercle bacilli, they developed hypersensitivity reaction when they were injected with tubercle bacilli or its protein, which is called as Coates' phenomenon. Now moving on to the second MCQ. Louis Pasteur is associated with all except vaccination of rabies, germ theory, pasteurization, vaccination of smallpox. So to this question, the answer is vaccination of smallpox, because they have asked except. So let us discuss about Louis Pasteur. Now Louis Pasteur is also known as father of microbiology because of his enormous contributions. So he is the one who described the fermentation. He is the one who also invented about the sterilization techniques, especially your hot air oven. Autoclaving steam sterilizer was developed by him, and he explained pasteurization of milk. Before germ theory, what was believed was theory of spontaneous generation of disease, but Louis Pasteur proved that no, that is wrong. Diseases are caused by the microorganisms present in the air, and it is not caused by a bad air or vapor, is what germ theory states, and this was given by Louis Pasteur. And coming on to vaccination, he discovered vaccines for rabies, cholera, and anthrax. So you remember it with the mnemonic CAR. So CAR. So he discovered vaccines for cholera, for anthrax, and also for rabies. Then what about vaccination of smallpox? Vaccination of smallpox was discovered by Edward Jenner. So therefore, this was discovered by Edward Jenner. So therefore, to this question, your answer is vaccination of smallpox. Moving on to the third MCQ. India ink staining is positive for which of the following? Pneumococcus, meningococcus, staphylococcus, gonococcus, enterococcus. So to this question, you have two options which are right. So one is pneumococcus and next is meningococcus. Now we will discuss why. Now basically when we talk about staining methods, so we have different types of staining. So the first one we have a simple stain. So where you just stain it with methylene blue or basic fission, and it gives the same color to almost all the bacteria in a smear. So that is called simple staining. The second you have is negative staining, which is usually done with India ink or nigrosin. So it is usually done with India ink or nigrosin. So this gives negative staining. So this is mainly to check for the capsule in a bacteria. And next you have got the impregnation methods, so which is to demonstrate thin structures like your flagella and spirochetes. Then you have got differential staining like your Gram stain, acid-fast stain, Albert stain. So all these gives different colors and this helps to differentiate different bacteria. And then apart from this you have special staining to stain your lipids or carbohydrates or flagella or spores. So all these are called as special stains. So now we are going to discuss about the negative stain, which is your India ink or nigrosin. So I told you this is to stain the capsule. So what is capsule in a bacteria basically? So sometimes you can see that the bacteria can be surrounded by a thick viscid structure or glycocalyx actually. So this thick viscid structure can be organized or can be unorganized. So when you are going to have it organized, the organized layer is called as capsule. And when it is going to be unorganized and loose material, it is called as the slime layer. So bacteria can have a capsule which is the organized glycocalyx layer, or it can have unorganized loose layer which is called as the slime layer. Some bacteria can have both capsule and slime layer. Say for example your Streptococcus salivarius. So this has got both capsule and slime layer. So example is Streptococcus salivarius. So this has got both capsule and slime layer. Now what are the capsulated organisms? So to remember this capsulated bacteriolist, I know it is tough. So let me tell you an easy way to remember. You remember it as best singers. So here you're going to use 2B. So best singers paid more, had costly pens. Best singers paid more, had costly pens. So here for C, you're going to use K. So best singers paid more, had costly pens. So where your B stands for Bacillus anthracis, the other B for Bacteroides fragilis, S stands for Streptococcus pyogenes, P for Pneumococcus, M for Meningococcus, H for Haemophilus influenzae, K for Klebsiella pneumoniae, and P for Pseudomonas aeruginosa. So this is how you remember the capsulated bacteria. You have capsulated fungus. So capsulated fungus is only one. So that is your Cryptococcus. So example for your capsulated fungus is Cryptococcus. So this completes the third MCQ. Now moving on to the fourth MCQ. Correct sequence in Gram staining. Your options are methyl violet, iodine, acetone, carbamazepine. methyl violet acetone iodine carbamazepine, methyl violet carbamazepine acetone and iodine, carbamazepine iodine acetone methyl violet. So here your answer is going to be methyl violet iodine acetone carbamazepine, which is your option A. So let us discuss about gram stain. So basically gram stain is a type of a differential stain. So this differentiates bacteria into gram positive and gram negative. So those that appears violet in color are termed to be positive. Those that appears pink in color are termed to be gram negative. Now how do you remember this order? You remember it as pilot in dark coat. So you remember it as pilot in dark coat. So this is easy way to remember the order of staining. So where P stands for your primary staining. So primary staining can be done by a crystal violet or gentian violet or methyl violet. So that is your primary staining. And second, it is going to be iodine. So it is going to be iodine. So N stands for iodine. And then D stands for decolorization. So decolorization can be done by acetone. So which you're going to do it for 1 to 2 seconds, or if you're going to use ethanol, it is going to be for 20 to 30 seconds. And you have to wash immediately. And after that, C stands for counter staining. So this counter staining can be with either carbamazepine or it can be with safranin. Now fifth MCQ, which is not present in gram negative bacteria. So your options are lipopolysaccharides, peptidoglycan, teichoic acid and all of the above. So yes, to this question your option is going to be teichoic acid. So when we talk about lipopolysaccharides, yes, it is present. Your option is like which is not present. But when we talk about lipopolysaccharides, it is present more in your gram negative. So therefore, no, this is not, because it is present in gram negative. Peptidoglycan, yes, it is present both in gram positive and gram negative, but the difference is it is thicker in gram positive and it is thin in gram negative. So therefore, peptidoglycan is also present. Though it is thin, it is present. And then coming on to teichoic acid. Yes, teichoic acid is present only in your gram positive bacteria. So it is not seen in gram negative. Now coming on to sixth MCQ, which is Craig's tube method differentiates motile and non-motile strains, virulent and avirulent strains, capsulated and non-capsulated strains, rough and smooth strains. So basically your Craig's tube method is mainly to differentiate your motile and non-motile strains. So it is to differentiate motile and non-motile strains. Now when you talk about a bacteria can move with the help of flagella. So it can move with the help of flagella. So based on the arrangement of flagella, your bacteria can be monotrichous, lopotrichous, peritrichous or amphitrichous. So which you all know. And this flagella, the presence can be demonstrated by direct and indirect methods. So when we talk about direct and indirect methods of demonstrating flagella, the direct methods include a tannic acid staining, so which is called as Leifson's method and Ryu's method. So you call it as Leifson's method or Ryu's method. So these are the tannic acid staining for direct demonstration of flagella. Next you have dark ground or phase contrast or electron microscope. Indirect method includes your Craigie tube method and hanging drop method, and you can use semi-solid medium like your mannitol motility medium. So like your mannitol motility medium can be used. So these are the indirect methods for demonstration of your flagella. Now based on what type of motility it possesses, you can approximately come to an idea what bacteria it is. If suppose there is going to be tumbling motility, then it states that it is going to be Listeria. If it is gliding motility, then it is because of mycoplasma. So your mycoplasma will exhibit gliding motility. Stately motility is going to be by Clostridium. So this is by Clostridium. Darting motility is mainly by two organisms. So one is your Vibrio cholerae and the second one is your Campylobacter. So your Campylobacter and Vibrio cholerae is going to have darting motility. Swarming motility is going to be by Proteus and also your Clostridium tetani. So it is also by your Clostridium tetani. Corkscrew motility, or you also call it as lashing motility, flexion extension motility. So it is by your spirochete, so which is commonly asked. So this is also an area of MCQ. So they just give you the swarming motility is seen in which of the following bacteria, corkscrew is exhibited by which of the bacteria. So this is also a potential area of a question. So MCQ can be asked from this area also. Now next MCQ, correct sequence of bacterial growth curve. Log phase, lag phase, stationary phase, decline phase. Lag phase, log phase, stationary phase, decline phase. Lag phase, stationary phase, log phase, decline phase. Log phase, decline phase, lag phase, stationary phase. So what do you think the answer is going to be? So basically when we discuss about the growth curve, it is going to be in this order. So you have lag phase, log phase, stationary phase, decline phase. So therefore the answer for this question is your option B. So how do you remember this? So you remember it with the mnemonic LSD. So here you are going to use 2L. So LSD, and these L's are going to be in alphabetical order. So it is going to be lag followed by log. So this is how you remember the bacterial growth curve. So the bacterial growth curve is in this order. It is lag phase, log phase, stationary phase, and then the phase of decline. So when I am going to draw the bacterial growth curve, so here I am going to take the time on x-axis, and here it is going to be the number on your y-axis. Now the curve will be like you have a lag phase, you have a log phase, you have a stationary phase, and then you have a phase of decline. So it is like that. So when I talk about lag phase, so what happens in lag phase? So in lag phase, there is no increase in number, there is only increase in size. So in your lag phase, only the size increases. But when we talk about log phase, it is also called as exponential phase. So therefore here the number is going to increase. So the number is going to increase. So what you can see over here is the number is increasing. So the number increases in your log phase. And in this stage, the cells usually will be small and they stain uniformly. So these are characteristic features of your log phase. Now when you talk about stationary phase, so what happens in stationary phase? Now usually in stationary phase, there is going to be the stationary count. So the count is not going to vary. But there will be gram variability. And there can be sporulation which occurs during the stationary phase. So these can be observed in stationary phase. So here there is no change in the number, but the gram variation is present and sporulation will occur. And finally, when we talk about the phase of decline, so as the name suggests, so it means that the count is going to decrease. And whatever involuted forms you can see, that can be seen in your decline phase. So in your stationary phase, you can see the sporulation, and in your, so S for S, so in your stationary phase, you can see sporulation. In your decline phase, you can see the involuted forms. So decline, so it is D. So therefore it is involution forms, which you can see in your decline phase. So this is about your bacterial growth curve. Now moving on to the 8th MCQ. Which of the following is microaerophilic? Your options are Pseudomonas, Campylobacter, Vibrio, bacteroids. So your answer for this question is going to be Campylobacter. Now there are different factors which is going to affect the growth of the bacteria, and one such factor which is going to affect the growth of bacteria is going to be oxygen. So your oxygen affects the growth of bacteria. So on basis of the oxygen requirement of a bacteria, you can either call it as an obligate aerobe, facultative anaerobes, microaerophilic, obligate anaerobes, aerotolerant anaerobe. So what is obligate aerobe? Obligate aerobes are bacteria which grows only in the presence of oxygen. So they very badly need oxygen to grow. So these are called as obligate aerobes. Say for example your Pseudomonas, tuberculosis, Bacillus, Brucella, Nocardia, all these are obligate aerobes. But when we talk about facultative anaerobes, so facultative anaerobes are those that can grow anaerobically. So they are aerobes but they can also grow anaerobically. So they are called as facultative anaerobes. For example, your E. coli, Staphylococcus aureus, so they are facultative anaerobes. They are basically aerobes but they can also grow in anaerobic medium. When we talk about facultative aerobes, it is just the opposite. They are anaerobes but that can also grow aerobically. They are called as facultative aerobes. So for example, your Lactobacillus. So Lactobacillus is a facultative aerobe. Now coming on to microaerophilic. So what is microaerophilic then? Microaerophilic bacteria are those which can grow in the presence of 5 to 10% of oxygen. So they require 5 to 10% of oxygen for their growth. For example, you have Campylobacter, you have Helicobacter and Mycobacterium bovis. So you remember it as CHM. So C is for Campylobacter, H is for Helicobacter and M is for your Mycobacterium bovis. What are obligate anaerobes? Obligate anaerobes is like they only grow in absence of oxygen. So that is why it is called as obligate anaerobes. So they grow only in absence of oxygen. If the bacteria is exposed to oxygen, then they die. So oxygen is lethal to these organisms. Say for example your Clostridium. So it is obligate anaerobe. Now what are aerotolerant anaerobe then? Aerotolerant anaerobes are those which can tolerate oxygen for some time but they cannot use it. They don't use it but they can just tolerate that oxygen. So they are called as aerotolerant anaerobe. Like for example Clostridium histolyticum. So it is going to be an aerotolerant anaerobe. So this is also an area where you can expect an MCQ. So which of the following is a microaerophilic, which of the following is an aerotolerant anaerobe, which of the following is obligate anaerobe. So this is also a potential MCQ area. Now 9th MCQ, true about exotoxins. LP is in nature, heat stable, not antigenic, toxoid can be prepared. So the answer over here is toxoid can be prepared. Now to know about this answer, we need to know what is the difference between exotoxins and endotoxins. So what are endotoxins and what are exotoxins? So when we talk about endo and exotoxins, endotoxins are basically lipopolysaccharides in nature and these are the part of your gram-negative bacterial cell wall and they are very highly stable. And when we talk about antigenicity, it is poorly antigenic. Because it is poorly antigenic, the neutralization by antibodies is going to be ineffective. And using endotoxins, there are no effective vaccine which is made available using endotoxins. So no effective vaccines using endotoxins till date. But on the other hand, when we talk about exotoxins just contrary, they are protein in nature. Unlike your endotoxin which is lipopolysaccharide, this is protein in nature and this is secreted both by positive and also your gram-negative bacteria. And next, when we talk about its stability, it is highly labile in heat. So it is heat labile. And because it is protein, it is highly antigenic. Because it is highly antigenic, neutralization by antibodies is also highly effective. And finally, you can have the toxoid forms prepared and which can be used as vaccines. So the toxoid forms using these exotoxins, yes, it is very highly effective as a vaccine. For example, your tetanus toxoid. So these are the main differences between your exotoxin and your endotoxin. Now moving on to the last MCQ, all of the following are true regarding endotoxins except neutralized by specific antibodies, heat stable, poorly antigenic, LPS. So they've asked except. So your answer over here after the previous discussion, you must be in a state to say that neutralized by specific antibodies. So this completes the first part of discussion on general microbiology. So we will discuss the second part next week. Thank you.